Hi everyone. In this session we're going to be looking at some more advanced assisted reproductive techniques and how else we can create pregnancies. So the assisted reproductive techniques that we're going to discuss in this session include artificial insemination, embryo transfer, embryo freezing or vitrification, ovum pickup and ICSI, embryo biopsy, oocyte transfer, semen sexing and cloning, and we're going to focus on these three, so embryo transfer, embryo vitrification and ovum pickup ICSI. So what are the advantages of assisted reproductive techniques? So these techniques, these ART techniques can help overcome limitations of reproduction, so whether it's mare or stallion subfertility, they can also just increase the productivity, so the number of offspring that can be produced from a certain animal can make the whole process more efficient. Assisted reproductive techniques can also reduce infectious disease risks. So because we're removing the need for natural breeding, we reduce the risk of sexually transmitted diseases between animals, and can also reduce injury risk to both the animals themselves and also personnel dealing with them. So collecting a stallion off a phantom reduces the risk to that stallion and also to the personnel involved. Assisted reproductive techniques also enable preservation and spread of genetic material of elite animals that aren't necessarily in the same geographic location or are not even necessarily still fertile or still alive even. So it massively sort of opens up the floor for possibilities in the world of reproduction. So just in order to set the scene, all the parts that need to function for successful conception and then embryo development and maintenance of pregnancy. So from the mare side of things, we need development of the egg. And then development of the sperm on the stallion side, resulting in fertilization, embryo development in the oviduct of the mare until the embryo enters the uterus, and then that pregnancy needs to be maintained in the uterus of the mare. So the most simplest form of assisted reproduction is artificial insemination, which we're just going to touch on. Most of you are going to be very familiar with this technique, so I'm not going to go on about it. But it's a very commonly used assisted reproductive technique. So the semen is collected from the male and deposited through the cervix into the female, and the semen can be processed, so either chilled and transported overnight or frozen indefinitely and used when desired. And can be transported over large geographic areas. Chilled semen is a very commonly used form of semen. Many stallions and mares are suitable for this, and the semen can be very successfully chilled and used with good results. So in general, if it's the stallion's first time that he's been collected and chilled ready for shipment, we often do a test cool before the start of the season to work out which semen extender suits that stallion best, and there's quite a lot of variation between individuals, so that way we can sort of maximise the quality of his semen being shipped. And most stallions in the right, when processed correctly, the semen will maintain good viability for at least 48 hours once it's been chilled, and so we can very, very happily ship overnight and have great success rates. And we monitor and manipulate the mare's cycle so that we can provide enough information for the optimum timing for the semen to be shipped relative to that ovulation. So frozen or cryopreserved semen, so this can last for decades without loss of viability. Once thawed, it does have a shorter lifespan than fresh or chilled semen, so generally just lasts 12 hours after thawing. So this means that we do need to have tighter management of the mare than with fresh or chilled semen, and so we need to know exactly when she ovulates, and ideally we inseminate within 6 hours of ovulation to maximise success rates. The other thing to say about frozen semen is that in the freezing process, some sperm will be damaged, and so the overall quality will be reduced. So it does only suit certain stallions, and we need to be starting with a really decent quality of semen to make to mean that once it's frozen, we still have good success rates. But it can be hugely successful for many stallions. So going back to this diagram, so for artificial insemination, basically all these parts must work. So we need normal egg development, we need normal sperm development. Those, once the sperm's deposited in the mare, we need fertilization to take place without problem, that embryo to develop, enter the uterus, and for pregnancy to be maintained. So moving on to embryo transfer. So this, again, many of you are very familiar with this, so we're not going to spend too much time on this, but with embryo transfer, the donor mare, or with conventional embryo transfer, the donor mare is bred, the embryo is then flushed from the uterus and transferred to a recipient mare who will then carry the pregnancy. The advantages of embryo transfer, so it increases the efficiency of breeding, so one mare can produce more than one offspring per year. And the donor mare doesn't need to carry the pregnancy, so she can continue in training if she's in a heavy competition schedule, and it can overcome certain donor mare subfertility problems. So particularly if she's perhaps old and has some endometrial fibrosis or degeneration, that means that she struggles to carry a foal to term. That can be bypassed because the embryo is going to be maintained within the recipient. Another advantage is that the embryo, once flushed, can actually be frozen and stored if desired, and doesn't need to be immediately transferred to a recipient, and that can be useful in some cases. And most breed registries will register an embryo transfer foal, obviously the exceptions being thoroughbreds, and also standardbreds, which only enable registration of one embryo transfer foal per season, so it does limit its usefulness there. And then back to this diagram. So although we need normal egg and sperm development and normal fertilization, and we do need the embryo to be able to enter the uterus via the oviducts, but we don't need the embryo or the pregnancy to be maintained in the uterus beyond 6 or 7 days, after which it will be transferred to the recipient. So the process of embryo transfer, so the donor mare is bred as usual using either fresh or chilled or frozen semen, whichever is desired, and the embryo is then flushed from the donor mare on day 7 or 8 after ovulation. So that flushing process involves flushing media being instilled into the uterus, so this blue line, through a cuffed catheter, which enables the uterus to be filled with fluid, and then the uterus is manipulated and to maximise the chances of this little embryo then being flushed out of the uterus through this bottom line here, out and through a filter which catches the embryo. So this is the filter here, and the embryo can then be identified under the microscope. The embryo is then washed and processed and then transferred through the cervix to the recipient, and the recipient will be synchronized to the donor so that she ovulated at a similar time to the donor, and has a hospitable uterus for that embryo. So the success rates of embryo transfer, so assuming that this is done by experienced people with a good technique, the success rate depends on the semen quality of the stallion, and the donor mare fertility or ability to produce an embryo and maintain that embryo up to day 7 or 8. Success also depends on the recipient mare fertility and the suitability of that mare. So, talking percentages, a donor mare typically produces an embryo 50 to 80% of the time, and that is dependent on her fertility. The rates of flushing, so if there is an embryo there, the rates of recovering that embryo are very high. When an embryo is transferred to a suitable recipient, on average, a pregnancy is established 70 to 80% of the time. Here at B&W, we actually have higher pregnancy rates for transferred embryos, so our pregnancy rates are greater than 90%. So choosing a suitable recipient mare, this is key to successful embryo transfer. So recipients should be young, so between 4 and 12 years old. They should have proven fertility and mothering skills, and they should be assessed prior to committing them to being a recipient. They should be assessed by a vet to determine that they are reproductively and physically sound. So the reproductive assessment may include tests such as uterine swabbing for culture and cytology, and potentially uterine biopsy as well. And the donor and the recipient, it's generally recommended that they're size matched, since the uterus that the embryo develops in largely determines the size of the resultant foal. So it generally makes sense to have the recipient a similar size to the donor. We recommend at least 2 recipients per donor in order to have the optimal recipient to transfer that embryo to. And the embryo can either be transferred to a recipient immediately if she's in the same location as the donor. The embryo can also be shipped to a recipient herd at an alternative embryo transfer centre without affecting success rates. So that's definitely another option. So when we're talking about flushing embryos, another option is to freeze or vitrify that embryo. And those frozen embryos can then be stored in liquid nitrogen indefinitely until they are transferred to a recipient mare. So vitrification is a technique that's a very rapid freezing process to achieve a glass-like state without the formation of ice crystals, which would damage that embryo. So why vitrify embryos? So embryo freezing enables preservation of genetics for holding and transfer to a recipient when it's convenient. So say a donor mare is bred and an embryo is flushed towards the end of the season. Transferring that embryo immediately would result in quite a late fall the following season, whereas by freezing it, we enable it to be stored over the winter and then transferred to a recipient early in the spring for an earlier fall. And it also removes the need for synchrony of the donor and the recipient. So if there isn't a suitable recipient at that stage, as the donor's mare is bred and flushed, then that's not a problem. Freezing embryos allows a worldwide market for selling embryos at auction. This is becoming increasingly popular, and there's an increasing demand for this. Here's just one advert for an embryo auction, and that in itself reduces the need for transport of live animals. In addition, something that's increasing the demand for vitrification is the fact that ovum pickup and ICSI, which we'll talk about next, or even just conventional embryo transfer, may result in more embryos than one owner desires in one season. So by freezing one or some embryos, that they can be spread out so that there isn't sort of a sudden appearance of many foals the following season. So the process of embryo vitrification, the donor mare is bred as usual, again using either fresh, chilled or frozen semen, and then that donor mare is flushed at exactly 6 days post ovulation, and that's really important because a really small embryo is needed for fast cooling rates and high success rates. So we need to know exactly when she ovulates and make sure that she's flushed at 6 days rather than the normal 7 or 8 days post ovulation. Alternatively, so larger embryos can be flushed later with decent success rates, but the fluid within the embryo needs to be removed before freezing, and that requires different equipment and more manipulation of that embryo. And so isn't straightforward, but as long as a small embryo at 6 days is flushed, then the success rates for vitrification are high. So in the freezing process, high concentrations of freezing media or cryoprotectants are used, and that minimises the damage to the embryo as it's frozen. So the cooling rates are very, very high for vitrification. So we're talking about so minus 15,000 to minus 30,000 degrees Celsius per minute. A reduction rate of that is that high. So the embryos are then placed in these specialised straws. So here's a sort of a diagram of one, so you can see where they sit at the bottom there, and then placed in liquid nitrogen, and they can be stored there indefinitely. When they need to be thawed, they then can be transferred to a recipient mare. And here's a microscope image of a frozen thawed embryo. And pregnancy rates following transfer of a frozen thawed embryo are generally reported at sort of 50 to 60%. So now moving on to ovum pickup and intracytoplasmic sperm injection, so otherwise known as OPU ICSI, which is definitely rolls off the tongue a bit more. So this is where this is basically egg or oocyte ovum harvest and in vitro injection of an egg with a sperm to produce an embryo. So basically test tube foals. So the first test tube foal was born in 1996 and produced in the United States. And since then, the technique's massively developed, and it's becoming pretty commonplace. And the reasons for doing OPU and ICSI, so there are several. So some mares may be unable to produce an embryo, particularly if they have ovarian problems or problems with the oviduct, so the fallopian tube, or potentially some problems of the uterus, and that may be the case in some older mares. And if we look at our diagram again on the right, so with OPU and ICSI basically bypasses a lot of those problems. So the egg still needs to develop and the sperm needs to develop, but we don't need fertilization to happen within the mare. We don't need the embryo to move through the oviduct or the fallopian tube, and we don't need it to enter the uterus or be maintained in the uterus. So it does bypass some of those problems. So mares with a history of abortion, those are sometimes ones that can be selected as suitable candidates. Also mares with a heavy competition schedule, and we'll talk about that a little bit more, but this can be really useful for those. Also because only a single sperm is needed, subfertile stallions can really benefit from this technique, or stallions either that may have already died or still be alive but have very limited semen reserves or frozen semen reserves. This can be really, really useful for these cases. So OPU, so the ovum pickup side of things, this is where we're harvesting the eggs. It can be performed in a standing sedated mare, and she can be at any stage of her reproductive cycle. So unlike embryo transfer, where we need to manipulate, follow her cycle, breed her at the suitable time, with ovum pickup, she can be at any stage of her cycle. So it can basically, and she doesn't need any hormone treatment. So it can basically sort of fit in with her training schedule, her competition schedule. We can just, if she has a week off here or there, we can say, okay, we'll book her in. The one thing that does really affect success rates is the number of follicles on the ovary. So ideally, the success rates are best when there are more than 15 small follicles present. So they're pretty small follicles, so less than a centimetre big, or less than a centimetre in diameter, but that definitely does increase success rates. So the process involves, so we have this ultrasound probe with a needle guide here, and this is put into her vagina, and then, so you can see it here, and then an arm is placed rectally to hold the ovary and bring it up close to that ultrasound probe, and then the needle is placed through the needle guide, and then visualizing using the ultrasound, the needle can then pierce the follicle here, and the fluid with the egg can be sucked back out through the needle, and this whole follicle can be scraped to maximise the recovery of those eggs. And we go through all the follicles on that ovary in the same manner to recover as many eggs as possible. So here are the recovered harvested eggs, or oocytes, and they're then filtered and transported to the ICSI lab. So that will be the lab doing the IVF or the in vitro fertilization. So once they reach the lab, they'll be matured and prepared, ready for fertilization. And the fertilization itself uses this micro manipulator. So there are these two needles. One holds has gentle suction and holds the egg still, and the other has a little needle that can draw up the sperm into it. So this side here, it's, this little irregularity there is actually the sperm. So it's drawn the sperm into it, and then the sperm is injected into that egg, and it's just the single sperm that does the trick. The injected oocyte is then transferred into a culture media and then monitored for development into an embryo, and that generally takes 7 to 9 days. And here we have an embryo here. And then that embryo can either be transferred immediately, fresh, into a recipient, or it can be vitrified and transferred at a later date, which is actually more common. So success rates to be expected with OPU and ICSI. So we expect to recover 60% of the eggs on the ovaries. So that's why it's important that the mare does have a decent number of follicles on her ovaries. If a mare comes in for OPU, for that cycle doesn't have a lot of appropriately sized follicles on her ovaries, we'll probably say, okay, it's worth just going home for a week or so, seeing what she develops, and then doing the OPU session when she has a few more follicles, and it will really increase the success rates. In general, we expect to produce 1 or more embryos every 1 to 2 OPU sessions. So it can be quite a lot higher, particularly if that mare has a lot of follicles on her ovaries. We can get quite a lot higher rates, but this is the sort of the average, the average expectation. When an ICSI embryo is transferred to a suitable recipient, a pregnancy is established 70% of the time on average. So then looking at embryo biopsy, so this technique enables diagnosis of certain genetic traits, so things like ovarolethal white syndrome, so this foal here is suffering from that, and quite a lot of muscle disease, genetic muscle diseases can be tested for through an embryo biopsy. Also determination of gender is possible. So the embryo is flushed at a standard time, so 7 or 8 days post-ovulation, and then shipped to a biopsy laboratory where it undergoes a biopsy and then can either be transferred immediately or frozen and transferred at a later date, depending on results of that biopsy. The biopsy process does not seem to affect subsequent pregnancy rates once transferred to a recipient, so there isn't significant concern over that. So another technique we have is oocyte transfer. So this is where eggs or oocytes are harvested from the donor mare, as for with OPU as we discussed, and then transferred to a recipient mare's oviduct or fallopian tube, and that recipient mare is then bred, and the donor's egg is fertilized. So it bypasses the fertilization within the donor, also embryo development within the oviduct. The embryo doesn't need to enter the oviduct, the uterus through the oviduct at all, and maintenance of pregnancy isn't required within the donor. But really, the success rates aren't great, and OPU and ICSI have really superseded this technique. Then moving on to semen sexing. So this is where we have a semen sample which the sperm within can then be sorted into X and Y bearing sperm prior to insemination to then determine the gender of the offspring. So the technology is based on the fact that that the DNA content of X and Y bearing sperm is different, and so it can be then sorted based on that using fluorescent stains. And the accuracy of sorting ranges from 90 to 96%. In the bovine industry, this semen sexing is widely used. It's very, very commonplace. But with horses, so as is often the case with ART and horses, they're special. There are difficulties, and these are particularly, there are very slow sorting rates with horses, given the kind of numbers of sperm that we're hoping for in a dose of semen. There's also damage to the sperm that occurs and does affect success rates and high cost. So advantages of semen sexing, of course, means that we're able to determine the gender of the offspring. Disadvantage, so the process does actually damage some of the sperm, and so reduces the quality of that semen sample. And so not all stallions will be suited, because their sperm may just not hold up to the process, and maybe result in a really poor quality semen at the end that doesn't get many pregnancies. This is sort of exacerbated by the fact that the numbers of sperm within these sex semen doses are actually really low, and the fact that the process is very expensive and generally not cost effective at the moment. And lastly, looking at cloning, so the first equine clone occurred in 2003, and the process involves basically DNA being taken from the connective tissue cell of a donor mare, and that DNA is then transferred to a recipient egg that's had its own DNA removed, and the recombined egg is then activated to then start developing into an embryo. So no sperm is required, and then that is transferred into a recipient mare that results in embryo. So here we have Tamarillo and Tomatillo. So the pretty famous clones from around these parts. So the offspring of cloned animals have identical genetic makeup to the donor, but they do have different mitochondrial DNA, and there are a variety of factors that affect the similarity between the offspring and the original donor, including this mitochondrial DNA. And so things like markings, so this is a bunch of clones here, and they don't all have the same markings. Also athletic potential, things like that are affected by many things, and they won't be identical to the donor. There are also a high incidence of placental and neonatal congenital problems in cloned animals, and there's a lot still to be understood. So it's really, it does have its place, cloning, but definitely is to be considered more a means of producing a breeding animal rather than kind of duplicating an athlete. So I think some big take-homes. So advanced assisted reproductive techniques are continuing to develop really rapidly, and it's a really exciting time in equine reproduction, and there's a lot of potential for them. And when used correctly, they can be really powerful and effective tools in increasing productivity, increasing success rates, increasing efficiency, and overcoming some limitations of certain animals. I think it's really essential to, before using these techniques, to absolutely consider very carefully the pros and cons of each technique, and how that corresponds to that particular case, that particular animal. And I think it's really important that everyone involved, owners, trainers, everyone has a really thorough understanding of the process, that particular technique, and a really thorough understanding of the expected success rates. And I think used carefully, I think that we can optimize the success, and have really great outcomes from these techniques. So if any of you have any questions, please don't hesitate to contact me or anyone on the stud team. My email address is on the first slide of this presentation. But yeah, definitely exciting times in the area of assisted reproductive techniques, and hopefully we'll be seeing lots of you this season. Thanks very much for listening.